So, as was mentioned, I'm here in Dallas, Texas, at the University of Texas Southwestern, and I'd like to give a little orientation, if you will, to who we are and what we do. So, the University of Texas Southwestern Peter O'Donnell Jr. Brain Institute Conquer Program is the full title of where I work and who I collaborate with. To give you a little geography, so within the United States, Texas is that very small region in the south. Dallas is in the northern part of Texas. You may have heard in the news the horrific flooding that's happening in the southern part of Texas along the Gulf of Mexico. Houston, Texas has been affected. We've got a lot of colleagues and friends and family down there. Thankfully, everyone's okay. The medical center fared actually relatively well, and things are back up and running within the medical center down there. Dallas, thankfully for us, was not affected by the flooding at all. On the side of your screen, you'll see a picture of the medical center in Dallas, and it spans more than a mile in a north-south direction, and about 3/4 of a mile in an east-west direction. It's made up of 4 major hospitals, the medical school, and a large research campus. And it's here within the Peter O'Donnell Brain Institute where I focus my work. As you'll see on the slide, the Conquer logo at the bottom is the group that I lead. The acronym Conquer stands for Collaboration on Neuroimmunology: question, understand, educate, and restore. And each of those words form the pillar of what we do. So this is a highly collaborative group with individuals from a variety of specialties. I'll point some of them out in a moment. We're focused on all conditions where the immune system attacks the brain or spinal cord or optic nerves. And our jobs are to question everything, do our best to understand. Our job is to educate and be educated. We learn a lot from you, our patients, and our families, just as much as hopefully we impart to you. And then obviously in the end, we want to restore function and restore protection against future events for our patients. And so I thought it'd be useful to go through a little bit about where we are with transverse myelitis. We've thought of it historically, and the way I've spoken about it for years, as a rare disease seeking a cure. As we're getting more and more savvy over these last few years, we realize we're wrong. It's actually a rare disease spectrum seeking various cures. And I'd actually like to spend a fair amount of time this evening talking about that spectrum, because what we've found in the last couple of years is the individual variances from patient to patient make up a huge reason for the variances in recovery, the variances in responding to different rehabilitation techniques, and the need to personalize the approach to every patient, because there are no 2 patients with this disease that are alike. And it's only when we stop lumping everybody together into one diagnosis that we'll actually start to make a lot of headway with this condition. So I'm going to get into the weeds a little bit with the science, and if there are questions, feel free to wave or throw something at the camera, and I'll pause and go into more detail. But I want to start with the classic description of this condition. And the classic description of transverse myelitis biology is that there's inflammation in the spinal cord. And what you're seeing on the screen over the word inflammation, all those little blue dots, are immune cells that have invaded the spinal cord of somebody who is diagnosed with transverse myelitis. So everywhere there's a blue dot, that's the immune system having infiltrated the spinal cord. Normally there wouldn't be any of those blue dots in the spinal cord. And those cells, those immune system cells, secrete a lot of substances that damage the protective coating around neurons. So by a show of hands, how many people in the room have heard of the substance called myelin? And keep your hands up if you've heard the term demyelination. Okay, so everybody's comfortable with this term. It's basically, if you think of it as the speaker wire of a stereo system, it's that rubber foam coating around the speaker wire. And when you lose that coating, the signal can't get from the stereo to the speaker as well, so the music doesn't sound as good. When we're talking about the human body, if those are the wires that are going to your right hand, if I lose the coating around the wires, my right hand doesn't work so well. If it's the wire going to my left leg doesn't work so well. So the symptom a person has depends on which wire gets affected. But what they all have in common in transverse myelitis is the immune system attacking the myelin and damaging that coating. Now, classically, we've described transverse myelitis this way, and we've said that if the inflammation is severe enough or is present long enough in the spinal cord, not only does the coating around the wire get frayed, but the actual wire itself can get cut. And we call this axonal transection. And the demyelination and the axonal transection, coupled with the inflammation, are what lead to all the symptoms, whether they be sensory loss, muscle stiffness, pain, bowel bladder issues, weakness, balance issues, whatever the symptom is. We have classically explained all of those symptoms by this damage: inflammation, demyelination, and then cutting of the axons. Well, it turns out that that's not 100% accurate when we're talking about transverse myelitis. And it's been growing over the last 4 years and really getting more clear in the last year and a half that this is only part of the story. And in order to go through the full story, I want to zoom out the lens and impart some new language. So by the show of hands, everybody in the room had heard the term myelin, and everyone had heard the term demyelination. So by the same show of hands, how many people here are familiar with the terms upper and lower motor neuron? So we have a few, okay? So not as many as the first one, which is what I usually find out. And then for my education in Australia, how common are discussions about the syndrome acute flaccid myelitis? By a show of hands, how many people here are familiar with this term and with this syndrome? So again, a few. So let me explain the difference between upper and lower motor neurons, and let me stress that if you get comfortable with the difference between these two pieces of anatomy, every question you ask about rehabilitation or symptom management will come back to this anatomy. All of it is based on this anatomy. And so getting comfortable with it will make a big difference. So this may make you feel like you're back in university or high school and nervous over the final exam. But indeed, this is actually the basis of all the care we give to our transverse myelitis patients. So what's depicted on the screen is a caricature of brain, and then the several slices of brain stem, which is the connection from the brain to the spinal cord. The three parts of the brain stem are labeled as midbrain, pons, and medulla, and then the spinal cord. Okay, and then you see...
Sorry, Professor Greenberg, Ross Duncan again here. We're just wondering whether you can maximize the PowerPoint screen. Just for this, just for it. Yeah, that's much better. Yeah, thank you.
Better. All right.
Thanks.
So, yep. So what you can see is the brain, the pieces of the brain stem, and the slice of spinal cord. And then you see a red line going out from the spinal cord to what's labeled as skeletal muscle. And let's actually see. Is that marking the screen for you?
Yeah.
Yeah. Okay. So the upper motor neuron is the wire that goes from the brain down the brainstem all the way to the spinal cord. The lower motor neuron goes from the spinal cord to the skeletal muscle. And those two wires are what are responsible for the contractions and the movement of every muscle in the human body, every skeletal muscle in the human body. But the physiology is very different. The function of these two wires is very different when it comes to motor movement. So if we keep the upper motor neuron and the lower motor neuron as separate, we can go through the different versions of transverse myelitis. So in the description that I gave you at the beginning, the classical description of transverse myelitis, what gets damaged in the spinal cord is the upper motor neuron, not the lower motor neuron. And so it's this upper motor neuron in the spinal cord that, when it's damaged, leads to weakness, difficulty moving a limb, and increased tone and stiffness and tightness. In acute flaccid myelitis, which is a variant of transverse myelitis, the damage is focused on this lower motor neuron right at its beginning in the spinal cord. So the brain, if you will, or the control center, I should say, of that entire neuron going out to the muscle lives right in the very front of the spinal cord. And that can get damaged, leading to death of this neuron and a very different pattern of weakness. Instead of having the stiffness, instead of having the hyperactive reflexes, when that lower motor neuron is damaged, you have atrophy and you have reduced reflexes in the muscle, and you'll start getting muscle twitching in a very different way, and you do not develop the stiffness, you do not develop the increased tone. Now, both of these cells can be damaged by inflammation in the spinal cord. So in one case, the inflammation might affect the upper motor neuron. In another case, it may affect the lower motor neuron, or it may affect both. Now, the implications are quite profound for keeping those two things separate. The first is relative to causation. So the way I described damage to the spinal cord, that very first piece of the triad was inflammation, those blue cells entering the spinal cord. And in classic transverse myelitis, where that upper motor neuron is affected and you have stiff muscles and tight muscles and loss of sensation and difficulty with bowel bladder function, the etiology is presumed to be autoimmune. The immune system gets confused, and instead of fighting an infection, it enters the spinal cord and causes damage to those upper motor neurons. Whereas when the focus of damage is the lower motor neuron, we know that the immune system can do this, but there's also ample evidence circulating over the last 4 or 5 years to say that some people have a virus that directly affects the lower motor neuron, directly affects the spinal cord right at that little part in the front, and that virus damages the lower motor neuron and recruits those blue cells in to cause even further damage. So the etiology behind classical transverse myelitis and the acute flaccid myelitis variant looks to be different. In one, there's a virus that begins the cascade within the spinal cord, and in the other, there is no virus in the spinal cord, it's just the immune cells invading the spinal cord. But there are implications also then for acute therapy. So when you're in the hospital with transverse myelitis, there are some therapies we give to get rid of those little blue cells, those lymphocytes that are in the spinal cord. And then there are other therapies that we may use to try and fight off viruses. So depending on which variant you have, we may pick different acute therapies. There are implications for what I deem natural recovery. And I want to spend a moment on this notion of natural recovery. And what I want to do, and I'm gonna shrink the slide for a moment because I'm going to watch your faces as I give this description, because I want to see if this is making sense. So for anyone who has been left with a deficit after transverse myelitis or spinal cord injury, the pathway to recovery has several pieces. For some people, they lay in bed at night and pray. For some people, they look at their arm or look at their leg and they yell at it and ask it to move and have these long negotiations with their legs trying to get them to move. For other people, they're in a pool and in a gym being yelled at by a physical therapist. For others, there are changes in diet. There are all sorts of ways we approach recovery. Natural recovery is when damage occurs to either the upper or lower motor neuron and the body instinctively engineers some recovery. So it can grow new myelin. It can compensate for dead lower motor neurons by having healthy ones sprout and take over the control of the skeletal muscle that was left behind, disconnected. So there are these natural mechanisms of recovery after spinal cord inflammation. The natural recovery mechanisms are going to be different to recover from a lower motor neuron injury than an upper motor neuron injury, because you need different types of repair. Now, do you grow new wires, either a new wire from the spinal cord to the muscle or a new wire from your brain down your spinal cord? All of the evidence we have found says, as human beings, we do not. We do not have the genetically encoded capability to grow a new wire from the spinal cord to the muscle or from the brain to the spinal cord. So whatever wires are left behind are the basis, the scaffolding, if you will, for natural recovery. Does that make sense? This notion of natural recovery. Now that is different, if you will, from rehabilitation. So how many people here, either themselves or a loved one, have ever taken part in rehabilitation or physical therapy? Hopefully this should be unanimous. I heard about the funds raised for the new center, including the pool and the hydrotherapist. I am happy to come down anytime and take part. I'm extremely jealous. Having a facility like that makes a very big difference. So everybody needs to take use of it. Otherwise, we will try and steal it and bring it here to the United States. So what are the implications for rehabilitation? So it turns out, and I'm going to enlarge this for you, it turns out that natural repair does some of the work for functional restoration. So everybody, or most people, assume that the way people get better after a spinal cord event is their body repairing. That's actually only part of the equation. A large part of restoration of function through rehabilitation is through plasticity. So shown here in the diagram, you'll see a couple wires, a couple upper motor neurons going from the brain down to the spinal cord. And in actuality, you have millions of wires heading down. And each of those wires does not encode a specific muscle contraction. Those wires get coordinated based on general movements. So when I stimulate one part of the brain, I don't just get a finger twitch, a muscle twitch, I get a whole limb movement in different directions. And so the connections between the upper motor neuron and then pools of lower motor neurons is quite complicated, but extremely useful from a rehabilitation perspective. Because it turns out in transverse myelitis, we almost never cut all of the upper motor neurons. That is extraordinarily hard to do, very difficult to do. And so when we're talking about transverse myelitis, it's a question of how many wires are still intact. So making up some numbers, let's say you had 1 million going down your spinal cord, and after those blue cells left, you were left with 500,000. Your job is to figure out a way to use 500,000 wires to get the job of 1 million done. And that's the notion of plasticity. And so rehabilitation is about getting your body to figure out how to get a job done with fewer wires dedicated to that job. And it's 100% possible. We see it all the time. We see people recover years, many years after their transverse myelitis, once we start approaching the rehabilitation in a very different way. And so a lot of it has to do with asking your body to recover. So when I talk about asking your body to recover, I don't mean sitting in bed looking at your legs and saying, please move. It turns out that your nervous system does not speak English. It turns out that your nervous system only responds to task-based requests. So if you want your legs to move, you have to try and move them. If you want your walking to improve, you have to work on your walking. And it doesn't just mean you work on your walking when your physical therapist is standing there watching you. It means that you work on your walk. You take the tools that your therapist teaches you, and you work on your walking every time you're walking. Because every time you take a step, your brain is trying to figure out how to use that connection. So if on Mondays and Thursdays in physical therapy, you do it correctly, great. But if on Tuesday, Wednesday, Friday, Saturday, and Sunday, you're doing it the old way, your brain is never going to learn. It's never going to figure out how to hardwire that compensation through the upper motor neurons. And so the implications for rehabilitation between an upper and a lower motor neuron are quite different. In a lower motor neuron, one of the implications has to do with our use of electrical stimulation. So this is a technique we use quite a bit with huge success, especially when dealing with upper motor neuron injuries, because I can take electrodes, place them over the skeletal muscle, apply a current, so the electrodes go on the skin over the skeletal muscle, and as long as there is a connection from a lower motor neuron to the muscle, when I apply the electrical current over the skin, the muscle contracts. And so if I want to train an upper motor neuron to work again, I can ask you to move a limb while applying the electrical stimulation and try and get that connection working again. What about if I've lost the lower motor neuron? Well, when I put the electrical stimulation device on, at least the current ones that we use, you don't get a muscle contraction because you need that intact connection between the lower motor neuron and the muscle to use most of the devices to get a muscle contraction. There are settings that we have learned to apply it to muscles that have lost the lower motor neuron in order to get them to contract, and it's changed the way we're doing rehabilitation. And so the approaches we take when there's a lower motor neuron and the approaches we take when there's an upper motor neuron for rehabilitation are quite different. There are further implications for outcomes research. So if I have somebody whose damage is from the upper motor neuron only, both upper and lower, or just the lower motor neuron, how I judge their recovery is going to be totally different. Why on earth is that important? As Ross was mentioning at the beginning in terms of funding for research and different things, if we don't know the outcomes of our endeavors, or if we lump together people who have totally different patterns of injury so that we get data that doesn't make sense, none of the work we do in transverse myelitis is ever going to pay off for a general population because we're not going to know how to measure outcomes. So we have to keep track of these different types of TM in order for the outcomes research to work. And then what most people are focused on is the restorative therapy. If I have a way to regrow myelin in the spinal cord, would it help somebody who had a lower motor neuron injury? No, not at all. It's not going to do a darn thing because they don't have that second connection. Likewise, if I have a way to restore the connection from the spinal cord to the muscle, is it going to help somebody who had an upper motor neuron pattern of damage? Not in the least, because that's not where they need the help. And so breaking people down into subsets is going to be incredibly important for doing the restorative research that we want to do. So with that in mind, I'm going to shift gears, and we only have a few more slides, and talk about the implication for TM and what we're doing on the research side. So at least in the United States, we quote about 1,500 cases a year. Now, it turns out that this has lumped together children and adults and all of the different versions of transverse myelitis, the classic idiopathic transverse myelitis, acute flaccid myelitis, and snuck in there are a few patients who will actually have multiple sclerosis but haven't been diagnosed yet. And when we look at the outcomes, the data is all over the map. We used to quote patients the notion of the rule of threes: a third recover almost fully, a third with some disability, and a third don't recover well at all. That number is a really bad statistic and probably not true, and it doesn't take into account any treatment interventions. We still, however, despite all of our treatments, do see variable outcomes. They're better than they used to be, but they're not the same. And when we look at our community, both in the U.S. and abroad, and Australia and Europe and Asia and Africa, what we find is no two people are alike. And so I find myself as a researcher moving between population level statistics and individuals. And when we're talking about transverse myelitis, we're talking about a lot of people. And these are all individuals with their own story. These are all individuals with their own experience. And what we have found in our work in the Conquer Project is the education is happening more from our patients towards us necessarily than us towards our patients. And what we found is we really have to listen closely because in learning about the differences between individuals, it's telling us a lot about what we need to be doing on the research side. And so the Conquer program, the Conquer TM program here at UT, we're here to provide care to patients, families, while we do research and while we treat and ultimately reverse the damage done by transverse myelitis. That's our mission. And how are we going about accomplishing it? So the collaboration. So I work with immunologists, I work with imagers, I work with surgeons, statisticians, psychologists, psychiatrists, radiologists, you name it, and everybody brings a different skill set to the table. And very important to us is a very robust circle of patients and families that we deeply collaborate with, because we found that when we lock all the scientists in a room to start hatching plans, often we get it wrong. And when we include patients and families in the process, we do a much better job. It's multidisciplinary. It's not just going to be a neurologist or a neurosurgeon who fixes this disorder. We're learning a lot from our colleagues in other disciplines. And one of the major things I've focused on while in Dallas is the integration of our missions. What I remind my patients is, obviously with consent, every visit is a research visit. Every time I see a patient, whether they're a month out, a year out, a decade out, 25 years out from their event, it's a learning opportunity for science. And we try to make sure that while we're providing clinical care, we're recording our experiences in such a way that we can learn from it in the future. And then we're trying to break the gridlock between the lab and the bedside. We're trying to accelerate the rate at which things can move from test tube and actually be a benefit to patients. And as I said, we partner with patients in all aspects of our mission. So what's the upcoming research that we can talk about? So there are a lot of projects going on, and I'm happy to take questions on all of them. The one that's probably going to garner the most attention over the next year has to do with restorative therapies. So the FDA, which is the U.S. Food and Drug Administration, it's our oversight committee over clinical research and drug-related research, has for the first time granted what's called an IND, an investigational new drug application for a stem cell that can remyelinate spinal cord. So we literally have been working with these cells for over a decade at this point. And in July of this year, the FDA granted the IND. What that means is we are not allowed to do human subjects research in the United States with a drug or a stem cell or anything until the FDA gives clearance. And as of the first week of July, they gave us our clearance. And so what you're going to see reported by the Transverse Myelitis Association in the next couple of weeks is that in the beginning part of 2018, we will perform the first in human phase 1 trial for remyelinating stem cells in patients who've been paralyzed by transverse myelitis. We're going to do what's called a phase 1 study. So this is a safety study where we're going to be taking 9 individuals who've been paralyzed by transverse myelitis and doing a surgical procedure where we directly inject the stem cells into the spinal cord. And these are stem cells that have been shown to regrow myelin in the nervous system. We've cured a lot of mice, and now it's time to take it from mouse to human and other animals besides mice, but mainly mice, and see if we can't recover function. So the way it works in the United States is there's 3 levels of trials: phase 1, phase 2, phase 3, and phase 1 is all about safety. So we're going to ensure that these cells don't, or the surgical procedure does not cause harm to the first patients who volunteer to take part in this research. So ever since I became part of the Transverse Myelitis Association and started doing work in this area, going back 15 years now or more, this has been our goal, to get to a place where we can start actually delivering remyelinating stem cells to the spinal cord. And we're excited to say that the next time we speak, I'll be telling you about first enrollments and how things are going. This is obviously an exciting project. It takes a lot of work. I can only show you part of our team. Everybody here has a different role to play, and I need about 2 or 3 more slides. But the nice part of our team is that we're not just clinician scientists or researchers or study coordinators. These are folks who get very involved in the community and try to partner up. For anyone who's been or watched the pictures online from the Transverse Myelitis Association camp that gets held every July or August. We go and we take part. This is from what's called the Messy Games, and it's self-explanatory. It's several stations where campers just get to attack each other and us with shaving cream, ice cream, pudding, you name it, and get as messy as you can. What I love about my team is they're not just committed to the science or the clinics, but they really are committed to the community as well, and we get a lot from taking part with events like these. And as Ross said, the TMA lost a giant in our field, Pauline Siegel, who passed away in a horrible accident this summer, just recently, and she's going to be very missed personally and professionally. For anyone here who ever had the absolute delight to meet Pauline, you know her smile was infectious, her love never ended, her grace was amazing, and she was really the key to the TMA connecting so many people, and frankly, for me being in this field, if the Transverse Myelitis Association and Pauline and Sandy hadn't done and what they did and weren't the people they were, I could have found myself working in all sorts of areas. But it's hard to get involved with a group like this and not just feel the passion that Pauline had and Sandy still has. And so she's going to be missed quite a bit, and we're going to be pushing forward to make her proud, because she's been waiting on the research a long time. So with that, those are just the updates and the slides I thought I'd go through. As I said, we could talk for several hours about different projects going on, but I wanted to leave time for any questions that you had. Happy to answer them. Hope this helped put things in perspective, especially when thinking about the symptomatic management and the rehabilitation. So with that, I'm happy to take questions.